Welcome back to this next video, and in this video we are going to talk about the bacterial pharyngitis. In the last video we discussed about the what is pharyngitis, and we focused on the viral pharyngitis. So to give you a brief overview of the last video, I've told you that pharyngitis, which is also known as the sore throat or a throat infection, that is actually a painful inflammation of the pharynx, which is the back part of the throat, and a pharyngitis that can involve some or all of these parts of the throat. That was the back third of the tongue, the soft palate, and the tonsils. And I've told you that this is actually the this is the pharynx, which is actually inflamed during the pharyngitis. And there was inflammation of the tonsil as well. These are the normal tonsils, these are the inflamed tonsils, and this was one of the images showing you the viral pharyngitis. In this particular video, we are going to focus on the bacterial pharyngitis, which is also known as the streptococcal pharyngitis, or sometimes known as the strep throat. Now, the first thing you have to do is to differentiate between the bacterial and the viral pharyngitis. How you can actually differentiate between the bacterial and the viral pharyngitis is there is a particular criteria, which is known as the FeverPAIN criteria, or the other criteria which is known as the Centaur Criteria, that can actually make you distinguish between the viral and the bacterial pharyngitis. So the first thing we have to do is we have to focus on this FeverPAIN criteria, how this is helping you in the identification of the bacterial pharyngitis. If you talk about this FeverPAIN criteria, there are certain things that you have to check for, and you have to check its absence or presence. Now, the first thing you have to check is fever during the previous 24 hours, the purulence, which is actually the pus on the tonsils, attend rapidly, that can include the pain and the fever within 3 days after the onset of the symptoms, the severely inflamed tonsils, and no cough or a coryza, which show you the inflammation of the mucous membrane in the nose. So in the FeverPAIN criteria, you have to check for these things. And each of the FeverPAIN criteria, it score 1 point. So there can be a maximum, the maximum score in the FeverPAIN criteria can be 5. Now, the higher score suggests more severe symptoms and likely bacterial cause. For example, if you are having fever during the previous 24 hours, that is going to give you 1 point. If there is purulence or pus on the tonsil, that means 1 plus 1 is 2. So higher the value, higher are the chances of the bacterial cause, or higher the chances of the bacterial pharyngitis. Now, this table is actually showing you the scores versus the interpretation in terms of the likelihood of isolating or presence of the streptococcus. Now, if the score is 0 or 1, that actually gave you a chance of 13 to 18% streptococcus or the bacterial pharyngitis. If the score is 2 or 3, that gave you a chance of 34 to 40%. And if it is 4 or 5, it gave you the chance of 62 to 65% of the bacterial pharyngitis. So as you can see, higher the score, higher are the chances of the bacterial pharyngitis. So this is one criteria which can actually help you differentiate between the viral and the bacterial pharyngitis. Now, the other criteria, that is known as the Centaur criteria, and just like the previous criteria, here you have to check for the presence or absence of something. Now, the first thing you have to check is pus on the tonsil. So if there is pus on the tonsil, that means you have to give it 1 point. If there is no pus on the tonsils, that means that the point over here, that will be 0. The second thing you have to check is the tender lymph nodes in the neck, the history of the fever, like over 38 degrees or 100.4 Fahrenheit, and the absence of the cough. So say for example, you have got pus on the tonsils, there are tender lymph in the nodes, so this is giving you 1 point, this is giving you 1 point. If these two are absent, that means the total score, that will be 2. If 3 are present, that means it is going to give you a score of 3. So in contrast to the previous criteria that we discussed here, the maximum score, it can be 4. In each of the Centaur criteria, each of these criteria, that is going to score a 1 point. Now if the score is 0, 1 or 2, that is giving you a chance of 3 to 17% of the bacterial pharyngitis. That means that if your score is 0, 1 or 2, that give you a very less chance of a bacterial pharyngitis. But if the score is high, like 3 or 4, that is going to give you a chance of like 32 to 56% of the bacterial pharyngitis. So these criteria, the Centaur criteria or the FeverPAIN criteria, can actually help you differentiate between the bacterial and the viral pharyngitis. So the topic that we are going to focus in this particular video is the streptococcal pharyngitis, or you can call it is the bacterial pharyngitis as well. Now this streptococcal pharyngitis, it is actually caused by the Streptococcus Pyogenes, which is also known as the group A Streptococcus, or for short it is known as the gas. So the Streptococcus pyogene, or the gas, that is actually responsible for causing the streptococcal pharyngitis. And again, streptococcal pharyngitis, that will be an infection of the pharynx, including the tonsils. Now this Streptococcus pyogene, it is placed in this group A Streptococcus, or it is called is the group A Streptococcus, because it have a specific Lancefield A antigen. Now this Lancefield A antigen, this is actually from a classification which is known as the Lancefield grouping. And this Lancefield grouping, it is actually a system of classification that classify the catalase negative and gram-positive cocci based on the carbohydrate composition on bacterial antigen found on their cell walls. So is the different bacteria, they will be catalase negative and gram-positive cocci. Within this particular group, the carbohydrate composition of the different species, they vary from each other. Therefore, the Lancefield grouping is actually classifying these catalase negative, gram-positive cocci into different groups based on their carbohydrate composition. So when you talk about the group A Streptococcus, there is a specific A antigen. Therefore, we call them is the group A Streptococcus. Now, if you split this this complete name of the Streptococcus pyogenes, that is going to give you some information about its, you can say, about its pathogenicity. Now, this strepto mean, this strepto from here, it actually means chain, and this coccus mean round. This means that these bacteria, they are going to make round chains. Now this pyo term, it refers to pus, which is actually the presence of the pus on the tonsils, and this genes actually refers to the formation. So these Streptococcus pyogenes are actually round chain-shaped bacteria which are responsible for the formation of pus on the tonsils. Therefore, they are known as the Streptococcus pyogenes. If you talk about the biochemical properties or the laboratory identification, if there is a suspected individual of bacterial pharyngitis, how are you going to identify the group A Streptococcus or the Streptococcus pyogenes? So the first thing you have to do is you have to go for the gram staining. And these group A Streptococcus or the Streptococcus pyogenes, they are gram-positive, and because they have a thick peptidoglycan layer, and when you stain them with the gram staining, they are going to give you a purple color. So in this diagram, if you see, they are making chains, and they are actually purple color when you stain them with the gram staining. Another important property of these bacteria, the Streptococcus pyogenes, is that they are non-motile in nature. You can use different kind of the media which can actually show you the non-motile nature of these bacteria. Now, how you differentiate between the motile and the non-motile bacteria? Now, if you grow the non-motile bacteria by stabbing them in a media, so their growth is confined to the stab line. You can see over here These are the non-motile bacteria. You have stabbed them in the media. So when you stab them in the media, their growth is actually confined to these stab lines and sharply defined margins. They are there. Surrounding medium is clearly transparent. As you can see over here, only there is growth in the stab, and the surrounding media that is transparent. And this is the uninoculated tube to which you can compare the motile and non-motile nature of the bacteria. If you talk about the motile bacteria, that actually give you a diffuse and hazy growth that spread throughout the medium, rendering it slightly opaque. So if you compare the negative and the positive one, you can clearly see the difference between the two. So this is the control, the uninoculated tube. If they are non-motile in nature, they are going to give you a growth like this, and if they are motile, you will be having a growth like this. So when you talk about the group A, Streptococcus or the Streptococcus Pyogenes, they are non-motile in nature. That means they are going to give you a result like this. Another important property of the Streptococcus Pyogenes is that they are non-spore forming bacteria, and they are facultative anaerobe. So this diagram is actually giving you some information about the aerobic and anaerobic nature of the different bacteria. If you talk about the obligate aerobes, you can see over here they are growing on the surface of the media. That means that they cannot survive in the absence of the oxygen. Therefore, they are called as the obligate aerobes. They can only survive in the aerobic condition. As against to the obligate aerobes, there are obligate anaerobes which cannot survive the presence of the oxygen, and they usually grow in the bottom of this particular tube, avoiding contact with the oxygen. If you talk about the facultative anaerobe, they actually prefer the oxygenated environment, but they can also grow in the anaerobic environment as well. So if you talk about the Streptococcus Pyogenes, they are actually facultative anaerobe in nature. Another important biochemical property is that they are catalase negative. They do not produce the catalase enzyme, which is actually responsible for hydrolyzing the hydrogen peroxide into water and oxygen, thereby giving you this bubble structure over here. So if you compare the catalase positive and the catalase negative organism, so if you add a catalase negative bacterial colony to hydrogen peroxide, you are not going to see any kind of the bubble. But if that is catalase positive, say for example the Streptococcus Pyogene, you are going to get a structure like this. This bubble formation is actually showing you a catalase positive result. This is another diagram showing you the catalase negative and the catalase positive organisms. Another important property of the Streptococcus Pyogenes is that they are positive for another enzyme which is known as the pyrrolidinoyl aryl midase enzyme. For short, it is known as the PYR enzyme. Now this PYR test is actually a rapid test which is used for the presumptive identification of the Streptococcus Pyogenes based on the activity of this PYR enzyme. Now what you do is that there is a disc which is actually impregnated with this particular compound which is known as the L-pyroglutamic acid beta-naphthylamide, that is actually impregnated into a test disc. And if you introduce that into the media containing the Streptococcus Pyogenes, the enzyme from the PYR enzyme from the Streptococcus Pyogene is going to act on this particular substrate, that is going to hydrolyze it, giving you another compound which is known as the beta-naphthylamine. Now when this beta-naphthylamine that is produced by the PYR, and you combine this beta-naphthyl with another reagent which is known as the PYR reagent, which is actually the p-dimethyl amino cinnamaaldehyde, that is going to give you a bright pink or cherry red color, as you can see over here. So if the organism, like in this particular case, if the Streptococcus Pyogene it is having this particular PYR enzyme that is going to hydrolyze this particular compound into the beta-naphthylamine, and when this beta-naphthylamine that is exposed to the PYR reagent, you are going to get this cherry red or this bright pink color over here. If an organism is negative for the PYR, that means this particular compound will remain intact. You will not be going to get any beta-naphthylamine. So if you expose this intact compound to the PYR reagent, you are not going to get anything, and this yellow color, that will be actually showing you the negative result for the PYR. So the Streptococcus pyogene, that is PYR positive, that is another important test that can help you identify the Streptococcus pyogenes. Another important property of the Streptococcus pyogene is their beta hemolysis. So before going into the beta hemolysis, I want to focus on different types of the hemolysis that is there. Now the first type of the hemolysis, that is known as the alpha hemolysis. And if you talk about the alpha hemolysis, that actually is this alpha hemolysis is produced by the Streptococcus pneumonia or the Streptococcus virulence, and this alpha hemolysis is also known as the green or partial hemolysis. Now, if you look at this green structure over here, if you grow a bacteria like the Streptococcus pneumonia or the Streptococcus virulence on a blood agar media, that is going to give you this green structure, this green color over here, and this green color means that the bacteria is having alpha hemolysis, or it is having partial hemolysis. When you talk about the other type of hemolysis, which is known as the beta hemolysis, which is actually a property of the group A Streptococcus or the Streptococcus pyogenes, what these beta hemolysis do is that, if you can see over here, you are going to get this yellow color on the blood agar, and this yellow color actually show that the hemolysis is complete. Therefore the beta hemolysis is also known as the complete hemolysis, that is going for the complete degradation of the red blood cells, thereby giving you a yellow color. So if you are getting a yellow color, that means that the organism is going for the beta hemolysis, or it is going for the complete hemolysis. So if you talk about the group A Streptococcus, they do the beta hemolysis by the help of a particular enzyme which is present in there, which is known as the streptolysin O, and this streptolysin O is actually giving the group A Streptococcus the ability for the complete hemolysis. Now this complete hemolysis, it can be done by another streptolysin, which is also known as the streptolysin S. But in case of the group A Streptococcus, the streptolysin, which is actually an oxygen sensitive cytotoxin, that is produced by most of the groups, most of the members in the group A Streptococcus, or by the group A Streptococcus. The third type of the hemolysis, that is known as the gamma hemolysis, and gamma hemolysis simply means there is no hemolysis. There will be no change in the color of the blood agar. You can actually see the streaks of the bacteria over here, but as these bacteria do not have the ability to go for the hemolysis of the red blood cell, so the color of the media, that is not going to change. And the gamma hemolysis, or the no hemolysis, is actually a property of the Staphylococcus saprophyticus or the Staphylococcus epidermidis. So the group A Streptococcus or the Streptococcus pyogenes, they are going for the beta hemolysis, or they are beta hemolytic bacteria, another important property of the group A Streptococcus. Now sometimes it is difficult to differentiate between the Streptococcus pyogenes and the Streptococcus agelacti, because both of them, they belong to the same genus, and most of their biochemical properties they are same. Therefore, in order to differentiate between the two, a specialized test is carried on, which is known as the Bacitracin test. Now this Bacitracin test, it is actually an antibiotic sensitivity test, and the Streptococcus pyogenes, they are sensitive to Bacitracin, and the Streptococcus agelacti, they are resistant to the Bacitracin. So if you look at over here in this particular plate, they are showing you the sensitivity of the Streptococcus pyogenes to the bacitracin. Any zone of inhibition that actually show the sensitivity of that particular organism to the bacitracin. So if you can see over here, this is the plate containing the Streptococcus pyogenes. You are actually seeing the zone of inhibition. That means that the Streptococcus pyogene is sensitive. If you talk about the Streptococcus agelecti, the same bacitracin disc, if that is applied, you can see clearly in this diagram that there is no zone of inhibition. That means that the Streptococcus agelecti is resistant to the bacitracin. So if you want to differentiate between the two on the media, if you are getting sensitivity against the bacitracin, that means you are having the Streptococcus pyogenes, and if you are having a resistance against it, then the organism that would be the Streptococcus agelecti. Now, there are certain important virulence factors in the Streptococcus pyogenes or the group A Streptococcus, and these virulence factors that actually enable the Streptococcus pyogenes to attach to the host cells, evade the immune response of the host, and spread by penetrating the host tissue layer. So the next thing we are going to discuss is the virulence factors of the S pyogenes. The first important virulence factor of the S pyogene is a capsule, which is the bacterial capsule in this particular case. Now, the bacterial capsule or the capsule of the S pyogene, it is a carbohydrate based bacterial capsule, and it is composed of the hyaluronic acid that surround the bacterium, protecting it from the phagocytosis by the neutrophils. Now, how these bacterial capsules they are actually preventing the phagocytosis of the by the neutrophils of these S pyogenes. If you talk about the capsule, this is actually the capsule. This particular rectangular structure over here, that is the capsule, and these are actually the different components of the bacterial of the cell wall, which are present the different component of the bacterial cell wall over here. Now, the glycoprotein, there are certain glycoproteins which are known as the endocytic pattern recognition receptor. If you can see over here, the endocytic penetration recognition receptor, which are present on the neutrophils. Now, what these endocytic pattern recognition receptor do is that they are going to recognize the pathogen associated molecular patterns present on the cell wall of the bacteria over here. If you talk about these pathogen associated molecular patterns that can include the peptidoglycan, the teichoic acid, the lipopolysaccharides, the mannins, and the glucans, which are present in many microorganisms. So what the endocytic pattern recognition receptor of the neutrophil do is they are going to recognize these pathogen associated molecular pattern, and when they recognize them, they are going to bind to them, thereby therefore going for the phagocytosis of the bacteria. Now, what the capsule do is that this capsule, they are going to cover the surface molecule, preventing the attachment of the endocytic pattern recognition receptor to these pathogen associated molecular patterns. And if these endocytic pattern recognition receptor cannot bind to the pathogen associated molecular patterns, that means the neutrophil cannot go for the phagocytosis, and the bacteria is prevented from phagocytosis, and it can actually spread in the body. Therefore, the infection that can also spread in the body. We will discuss about other virulence factor in the next video. So if you like the video, press the like button, subscribe to my channel, and share it with your friends.